Hello students, myself Dr. Vinita Rathore, and this is my fourth video on general anatomy, functions and histology of digestive system. In this video, we will discuss about the general anatomy, functions and histology of small intestine and large intestine. The word intestine is derived from Latin word meaning internal. The two intestines, that is, small intestine and large intestine, nearly fill all the interior of the abdominal cavity, and that is why they are called as intestine. Small intestine. Small intestine extends from pyloric sphincter to ileocecal wall, where it empties into large intestine. It occupies a significant portion of the abdominal cavity. It is thin-walled tube which measures about 6 meters in length. It is the longest section of the digestive tract. It is actually narrower than the large intestine, and that is why it is called as small intestine. It is coiled because it has to occupy the abdominal cavity and it is very long. So it has to coil to fit into the abdominal cavity. The food which comes from the stomach in the form of chyme enters into the small intestine. Small intestine is the place where chemical digestion completes, and the ingested nutrients have to spend at least 12 hours in the small intestine to be completely digested. Small intestine is responsible for absorption of most of the nutrients which are digested in the alimentary canal. Anatomy or regions of small intestine. The small intestine is subdivided into three regions. They are the duodenum, jejunum, and ileum. The duodenum. It is the first and the shortest segment of the small intestine. It measures approximately 25 centimeters in length. It originates at the pyloric sphincter, and it is having a major duodenal papilla, which is the part where the main pancreatic duct opens into the duodenum and the pancreatic juices from the pancreas are released in this part of the small intestine. The second part is the jejunum. It is the middle region of the small intestine. It is approximately 2.5 meters in length. It makes up approximately 2/5 of the small intestine's total length. It is the primary region for chemical digestion and nutrient absorption. There is no clear demarcation between the jejunum and ileum. The ileum, which is the last and the longest part of the small intestine, it measures about 3.6 meters in length. It forms approximately 3/5 of the small intestine. It terminates at the ileocecal valve. It is provided with sphincters which controls the entry of materials into the large intestine. This diagram shows the anatomy of small intestine, in which we can see duodenum, which is the first part of the small intestine, and it is now connected with the jejunum, which is coiled part, and the last part, which is called as the ileum. All these three parts of the small intestine here are surrounded by large intestine. Histology of small intestine. The wall of small intestine is composed of the same four layers typically present in alimentary canal, that is mucosa, submucosa, muscularis and serosa. Three features of mucosa and submucosa are unique in small intestine. They are the presence of circular folds, villi, and microvilli. Circular folds or the pleci circularis. These are the circular pleats in the mucosa and submucosa around the interior of small intestine. These circular folds slows down the movement of food because they are circular in shape and causes chyme to move spirally, which slows down the movement of chyme and provides the time needed for nutrients to be fully absorbed. These circular folds increase the surface area of the intestine. The second feature is presence of villi. There are 20 to 40 villi per square millimeter of circular folds. These are minute finger-like projections. They contain blood capillaries which are composed of one arteriole and one venule and lymphatic capillary called lacteals. They also further increase the surface area of the small intestine. The third feature is presence of microvilli. There are approximately 200 million microvilli per square millimeter of small intestine. They are submicroscopic in size and they are present as projections on single cells. They also aid in absorption of the nutrients by small intestine. Intestinal glands, which are also called as crypts of Lieberkühn, they are present in the intestine and they secrete intestinal juice, which is slightly alkaline in nature, the pH of which ranges from 7.4 to 7.8. Approximately 1 to 2 liters of intestinal juice is secreted daily. These intestinal glands are also responsible for cell regeneration. This picture shows the histology of small intestine. Here you can see the simple columnar epithelium which is provided with microvilli and these microvilli absorbs the nutrients. Here you can see intestinal glands and goblet glands. Both of these intestinal glands and goblet glands are situated in the epithelium and they are helpful in various functions. The goblet cells are responsible to secrete mucus while the intestinal glands helps in the digestion and absorption of nutrients in the chyme. There are enteroendocrine cells which are responsible for secretion of hormones which initiates the activation of various enzymes in small intestine and thus they allow the digestion into the small intestine in alkaline medium. Large intestine. Large intestine is the terminal part of alimentary canal which frames all three parts of small intestine. It runs from appendix to anus. It measures about approximately 1.5 meters in length and 6.5 centimeters in diameter. Despite it is smaller than small intestine, it is called large intestine because it is more than twice the diameter of small intestine. Its primary function is to finish absorption of nutrients and water. It absorbs most of the water and electrolytes from remaining digestive material. Watery material that first enters into the large intestine soon solidifies and becomes feces. Stored fecal material until the body is ready to defecate. When it is filled with fecal material, time to time it removes the feces from the body. It helps in the absorption of very small percentage of the nutrients which are still present in the digested material. It, it is composed of four segments: the cecum, colon, rectum, and anal canal. Anatomy or regions of large intestine. Large intestine is subdivided into three main regions, namely cecum, colon, and rectum. The ileocecal valve, which is present between ileum and large intestine, controls the flow of chyme from small intestine to the large intestine. Cecum. Cecum is the first part of the large intestine. It is a sac-like pocket at proximal end with appendix. Appendix is a winding tube that attaches to the cecum. Appendix is a vestigial organ. The second part of the large intestine is colon, which is further subdivided into four different regions. The first is ascending colon. Ascending colon is present on the right side of the abdomen between cecum and right colic flexure. The second part is transverse colon, which is present horizontally. The third part is descending colon, which is present on the left side of the abdomen between left colic flexure. And the last part is the sigmoid colon, which is S-shaped in structure and it bends near the terminal end. The final part of the large intestine is the rectum, which is the terminal part of large intestine and it ends in anal canal. The rectum is provided with two sphincters, namely the internal involuntary sphincter and external voluntary sphincter. The internal involuntary sphincter is made up of involuntary muscles, while the external voluntary sphincter is made up of skeletal muscles. Except when defecating, both these sphincters usually remain closed. In this picture, you can see all different parts of large intestine, including cecum, colon, and rectum. Histology of large intestine. Histologically, large intestine has several notable differences with that of small intestine. Mainly, circular folds and villi are absent in large intestine. The first layer, which is mucosa, this mucosa is having abundant goblet cells which produce mucus. The mucosa layer is made up of stratified squamous epithelium near anal canal. Goblet cells are also present here, which secrete mucus that eases the movement of feces and protects intestine from the effect of acids and gases which are produced by enteric bacteria. No villi are present in large intestine. In the muscularis layer, we can see that longitudinal muscle layer is incomplete and it forms three bands of smooth muscles which are called as tenecoli. Tonic contractions of tenecoli bunch up the colon into a succession of pouches which are called haustra. The third is circular muscles which forms pockets or the haustra between bands. These haustra are responsible for wrinkled appearance of colon. This diagram shows histology of large intestine. In this picture, we can clearly see all the four layers of large intestine, namely mucosa, submucosa, muscularis, and serosa. Goblet cells and Intestinal gland cells are also visible here. These cells are responsible for secretion of mucus, which prevents the large intestine from damage from the undigested food material. So with this, we complete the entire topic of general anatomy, functions and histology of digestive system. In the next videos, we will discuss about accessory digestive organs.